actual organisms, not viruses, which aren't organisms, but if you just replace the word microorganisms with pathogens, and not so much culture-based diagnostics, essentially the format is the same as we started talking about viruses. So we're trying to learn about them, their characteristics, and the diseases that we associate with them. Alright, we did overview of viruses a little bit in block 1, so we'll do a bit of a refresher before we get going in any kind of detail. Some major characteristics, absolutely obligate intracellular parasites, unlike even some of the obligate intracellular parasitic bacterial species that we've talked about. They at least have some of their own synthetic pathways, but in terms of viruses, obligate. And parasites, I mean, they completely parasitize as host cells, so not a lot of mutualism or benefit there. And this seems like an obvious point to make, that they have to be infectious in order to endure in nature. But they do go out of circulation, and viruses do become extinct. So if they, if they lose their host base or something like that, they go out of circulation. Or if they're not replicatively competent anymore, they go out of circulation. Cannot make energy, again, they don't have those synthetic pathways, they don't have their own ribosomes. Like bacteria have their own ribosomes, so they can do their own protein synthesis. These cannot, and that's key. They may or may not rely on host cell polymerases, depending on their own genome and their own genome requirements. And because of that reason, it's very difficult to develop antiviral drugs if the viruses are using all the host cell machinery. It's not like we can develop an antiviral drug that's going to target like their ribosomes, because their ribosomes are our ribosomes. So it makes it a little bit harder. We have to look at things that allow us to target the viruses themselves, so we can get that selective toxicity that we talked about a little bit in block 2. So the genomes can be either RNA viruses or DNA viruses, but not both. If you have an RNA virus, one of the implications, there are several implications, is that you need to do like a PCR for these viruses. You have to reverse transcribe that RNA into DNA so you can do a PCR. But there are lots of other implications and reasons why it's good for us to understand the basic biology of viruses. That's just one example. We also talked about, in block 2, when we were talking about microbial genetics, we were talking about the central dogma of molecular biology, or central dogma of life, where you have like a DNA genome in cellular life. Genes get transcribed, those transcripts, those mRNA transcripts get translated into proteins, and then the genome itself gets replicated. Viruses, they violate the hell out of the central dogma of life, because they can. They're viruses, so they do some funky things. RNA viruses have RNA genomes, and if those RNA genomes are in the right orientation or polarity, they can be translated directly into proteins. Weird. Wow, some weird animation on here. If it's a negative sense virus, which we'll talk about what that means in more detail, then it has to transcribe a positive sense copy of itself that can be translated into proteins first. So if I'm confusing the hell out of you right now, that's fine. We'll get that pretty heavy today. And then lastly, another category that violates the central dogma are retroviruses. Retroviruses like HIV are technically positive sense RNA viruses, but they're the ones that integrate into the host cell genome. So they use reverse transcriptase types of polymerases to make a DNA copy of themselves that can then be integrated into the host genome and establish long-term persistent infections. And from there, once that DNA is integrated into the host genome, the host treats it like any other piece of DNA. If there's a gene, and if that gene is infringed, it just transcribes that gene and translates it into protein. Except that's now a viral protein instead of a host cell protein. Lastly, the viruses will rely on as much of the host cell machinery as they possibly can for their viral infectious cycle. But one thing that the host cell cannot do is assemble virus particles for them. So they self-assemble. The host cell is designed to do that. We haven't evolved to put viruses together, so they have to do that on their own. And these components have the ability to self-assemble into progeny virus particles themselves. And similarly, on that note, when we talk about cellular life, we use slightly different terminology for the way that organisms have to reproduce. growth division that we would use to talk about binary fission for bacteria or mitosis for eukaryotes, not appropriate for viruses. There's no growth, there's no expansion of the macromolecules that compose cells with this. Rather, you just get a template copy of the genome, and that template is amplified. There's subsequent copies of that made, and then the proteins are amplified as well. So from one starting input of a virus, you can get tens or hundreds of output copies, but there's no growth per se. It's more like an assembly or a factory. And then lastly, in terms of semantics, a lot of people say killing viruses or the virus will die, and that's fine as long as you guys have your understanding that that's not really the right precise terminology to use. You can't kill something that's not alive. So it's fine to say that colloquially if you're talking about killing viruses, but in your mind, the way you think about these things, your understanding should be a little bit more involved. And that you can inactivate them or destroy them or render them non-infectious. Those would be accurate ways of describing getting rid of viruses, but not killing them. In terms of the taxonomy of viruses, in the field of microbiology, all of these different kingdom have taxonomies that adhere to Carolinian system of classification. So you have that genus and species name where we talk about an organism that we've become familiar with, like Streptococcus pneumoniae, for example. And then like us as humans, Homo sapiens, genus, species, same nomenclature. When you're talking about viruses, there have been attempts to conform viruses to that Carolinian nomenclature, but so far those have failed. There's a body called the ICTV, the International Committee on the Taxonomy of Viruses, that occasionally attempts to standardize viral names according to this two-naming Carolinian convention. So far, we've not been able to do that, but maybe someday eventually we'll start calling viruses by like a genus species name. However, so they're not on the tree of life, they don't fit into that convention. However, we do try to organize them, so we do have like a rough equivalent of how we would categorize viruses on a kingdom, phylum, order, class, family, genus, species, normal, taxonomic, hierarchical progression. We try to do that as well. An example of that is like influenza virus, right? We all know about influenza virus, which is what we're talking about today. That would be at a family level, so it's an Orthomyxoviridae. That suffix on viridae denotes that it's at a family level. The lower tax underneath that would be at the genus level, where we're talking about influenza A, influenza B, influenza C, etc. So it kind of loosely conforms to that system. In terms of structure, we'll talk just in terms of the brief outline of the commonalities in virus structure, starting with the genome itself. As I mentioned, you can have either a DNA genome or an RNA genome. And even within those two broad brackets, those nucleic acids can be either single-stranded or double-stranded. So the shorthand there is DS for double-stranded or SS for single-stranded. You can have single-stranded RNA, which a lot of viruses are, a lot of the ones we're going to be talking about. Double-stranded RNA, which very few are, just rotavirus in terms of clinically important viruses. And then for the DNA viruses, a great number of the DNA viruses are double-stranded DNA. So they behave basically like our own genome. A small percentage are single-stranded DNA viruses, things like parvovirus for dogs, particularly this virus that affects dogs, can also affect humans. And then for the RNA viruses, these have a further classification into whether or not they're positive sense or negative sense. Again, a little bit more detail about those classification schemes later. But that has implications for how they replicate in the host cell. So that's the genome. In terms of how these genomes are packaged, all viruses need to have their nucleic acid genomes, whether it's DNA or RNA, protected by some sort of protein coat. That's called the capsid protein shell. And how that protein shell interacts and protects with the genome makes it either helical symmetry or an icosapentagonal symmetry. If it's a helical symmetry, like this figure in this cartoon in the middle there, those nucleocapsid proteins are binding more or less directly to the genome itself. This is mostly going to be a feature that you find in RNA viruses. But what that does for the virus is protect it as it uncoats in the host cell. Naked RNA, in particular, doesn't last very long. It's a transient molecule, so it needs some sort of protection. So you only kind of get uncoated or undressed, if you will, very briefly, and otherwise you need to be protected by protein or you're going to be degraded by RNases, enzymes that degrade RNA. Icosahedral symmetry, this can be found in DNA or RNA viruses. That would be more like this top figure, icosahedral geometry, or the bottom figure, either one, but the capsid is more loosely packaged around the genome, so there's space in between that, the nucleic acid genome and that capsid shell. And then lastly, whether or not a virus gets a secondary exterior that it derives from the host cell in form of an envelope, a lipid envelope. They do not make their own. The capsid is viral, so they have to encode their own genes for this viral capsid. But when it comes to an envelope exterior, that's something that they pick up as they're budding out of usually the plasma membrane of the host cell, but sometimes the Golgi, sometimes the endoplasmic reticulum. So that's host cell derived. A lot of viruses are envelope viruses, and a good number of them are not. The ones that are not, we call non-enveloped, or capsid, or naked. You'll see it in various ways, but the bottom line is there we have a protein exterior. In terms of implications as to whether or not these have an enveloped exterior or a capsid or non-enveloped exterior, start with the enveloped ones first. If you are just protected by that lipid envelope layer, you're not going to last very long as a general rule out in the environment because it's a lipid bilayer. So these tend to be more fragile viruses, and that has implications for how they're transmitted. An example, if you think about some of the bloodborne viruses, what kind of viruses do you think of with bloodborne viruses? HIV, hepatitis B, hepatitis C. So those are envelope viruses. They're transmitted by parenteral transmission, so blood from person to person. They don't have to linger out in the environment. If they were to, they would be desiccated or they would be susceptible to drying, heat, UV, things like that. They would be destroyed. So transmission is one of the big factors that's affected by whether or not a virus is enveloped or capsid. These viruses usually have to stay wet. A lot of respiratory viruses are enveloped, so they're transmitted in nice wet aerosol droplets from person to person. And as you can see up here, this is an electron micrograph of a host cell where you're starting to see in a 4-panel display a virus that's beginning to bud from a host cell. And then as it slowly becomes untethered and pinches off, it's just like it's taking a little bit of that skin of the virus with it. Not a big deal for the virus itself, but if you're talking about hundreds or thousands of viruses budding off from a host cell, that gets to be very much injurious to the host cell to lose that much membrane. Oh wow, she wouldn't animate these. In terms of capsid viruses, just very quickly, what's exposed again on the outer side outer exterior of that virus is a protein capsid, viral protein capsid. So protein is a more robust macromolecule than lipids, and these do better when they're in the environment. In terms of transmission, if I'm infected with a capsid virus, non-enveloped capsid virus, and I'm shedding it, it can actually survive on surfaces for foam-like transmission for considerable periods of time. Some of them hours, some of them days. So important to kind of conceptualize that. Also, for these viruses, when they're infecting host cells, the way that they leave host cells is a little bit different than envelope viruses. Envelope viruses can be released from host cells without immediately killing that host cell, because you just have this gentle letting off mechanism, right? The host cell survives from that. But with capsid viruses, they don't get released from the cell by pitching off and blooding off. Rather, they build up en masse in great numbers, and then all of a sudden the cell can no longer stand the number of viruses in that cell, and it bursts. So there's lysis, and when those cells lyse, they release a huge number of progeny capsid viruses. So different mechanisms of release between envelope viruses and non-envelope viruses. These also tend to be, in addition to like the transmission implications that I was talking about, water transmission in the environment. If we have viruses that can be like GI viruses, like norovirus, rotavirus, in order to withstand the pH of the stomach, usually those viruses are going to be capsid viruses because the acid in the stomach would tear envelope viruses apart. That's another implication there. Yeah.
With the enveloped viruses, since they have that additional layer, would they still have that protein layer underneath the lipid membrane and glycoprotein?
Yeah, exactly.
So why would they be more susceptible?
That's a great question. Okay, so he's asking if you still have an icosahedral symmetry underneath that lipid bilayer, why wouldn't the virus survive injury or destruction of its envelope? Any thoughts on that? Yeah.
Like a shearing kind of thing, shears the
Possibly, but what about, so less directly in terms of survival of the virus? Let's say you are washing your hands and you know that mechanically soap molecules will tear an envelope apart, right? That's what it does. But let's say it doesn't destroy the capsid on the inside. What happens is this cartoon is showing you these little like viral glycoproteins on the surface of them. That's how the virus enters and infects a new cell. If you destroy that, those viruses can no longer infect new cells. So what do we say about viruses needing to be infectious in order to endure in nature? What you've done is destroy that virus's ability to infect cells. Whereas if a virus is designed or evolved to have its capsid proteins with these glycoproteins on the surface, it can still get in and infect cells. Okay, but that's not the case with envelope viruses. Their receptors are on the outside, on the viral membrane. Okay, oh my gosh. Okay, for DNA viruses, we talked about characteristics of the X-ray or whether or not it's enveloped or non-enveloped. Now we're going to talk about the characteristics of DNA viruses versus RNA viruses. When you guys are studying this material, try not to conflate the two together. They're different properties, so it's important that you keep them separate in your head. For DNA, we know as a molecule, it's a more stable molecule. So DNA viruses like the herpes viruses or hepatitis B virus or some other viruses, they tend to be more stable. They're not as easily destroyed. They're also the ones that are able to establish persistent or latent infections, which we'll talk about in a couple slides as well, because RNA is more a temporary molecule. So when a DNA virus infects a cell, like HIV, once it reverse transcribes and has that complementary DNA strand, that can be persistent in a cell, persistent for decades. But an RNA virus tends to have more acute infections. You know, get in, get out, do your business, acute infections. In terms of the molecule that is recognized by the host cell RNA polymerase recognizes that viral DNA like its own. There's nothing to tell it that it's something different. So inherently, it basically behaves like host cell DNA in terms of transcription and replication. That is different than RNA viruses. And because of that, because all the machinery it needs for replication is in the nucleus, these viruses, once they get into the host cell, they have to be transported, at least the genomes have to be transported into the nucleus where it can access the DNA polymerases for replication and the RNA polymerases for transcription. And then lastly, this is true of many viruses, that transcription is temporally regulated, meaning that there are genes that have to be transcribed initially first, usually enzymatic genes that are important for replication, and then later genes tend to be things that are important for building the viruses, like structural proteins. So there's a sequence, it's very tightly controlled and important for that. In terms of classifications, this will kind of come together later, but here we see an example of a DNA virus. It's a pox virus called smallpox. Briefly talked about it before. But it's just a double-stranded DNA virus. So once it gets into the genome, those transcripts they come out because the RNA polymerase are transcribing directly from the viral DNA to synthesize new particles. In terms of RNA viruses, RNA molecule, labile, transient, think about the RNA species in our cells, ribosomal RNA, messenger RNA, transfer RNAs, all of those are designed to be temporary, they don't last long. So ditto with RNA viruses. They replicate out in the cytoplasm, they usually don't have any business or need to get into the nucleus. But when you think about the way RNA might be replicated in a way that doesn't happen in a eukaryotic cell, you would realize that the host cell does not provide polymerases that are capable of making RNA copies from RNA. We transcribe from DNA, but we don't use RNA-dependent RNA polymerases. So, viruses have to bring their own RNA-dependent RNA polymerases. They have to package that in that virus particle. Let's see, genome structure and clarity determine how viral mRNA is generated and proteins are expressed and processed. That's sort of cartooned out here in some other examples of viruses. These are RNA viruses. If we look at this positive sense RNA example, we're talking about coronavirus like SARS-CoV-2, for example. That's a positive sense RNA virus. When that cell enters when that virus enters the host cell, that positive sense strand of viral genome is in the right orientation to be directly translated into protein. Those are the implications there. However, you still have to make an intermediate. This is called an antigenome, so that's a negative sense strand. And then from that, antigenomic copy, this is a temporary molecule. You can go and make many more genomic copies that end up getting packaged in the virus. So the genome itself is positive sense, the temporary copy is negative sense, and then the lasting copies that get packaged into the new viruses are positive sense. When you're talking about negative sense RNA viruses, the reverse is true. It's a negative sense genome, you have to have a positive sense intermediate antigenome, and then negative sense genomic copies that get packaged. In terms of transcription and translation, that negative sense RNA molecule is in the wrong orientation to be recognized by host cell polymerases. So you have to first transcribe or replicate a positive sense thread, and then that can be used as a for translating of new viral proteins. Last point on this slide, well, second to last, most native RNA viruses are enveloped, just kind of like an observational thing. And then lastly, RNA viruses have a higher mutation rate than DNA viruses. Why do you think that is?
RNA polymerase.
Yeah. Yeah, the RNA virus is crappy, it's low fidelity, so more transcription errors, more replication errors, and not as many proofreading capabilities as DNA polymerases. You can kind of see this in this slide a little bit when we compare genome size and actual organisms, cellular life, to viruses in terms of the mutation rate errors that occur. On the x-axis here, we're looking at genome size. We would fall into this category of higher eukaryotes. Our genome, the human genome, is in the order of around 3 billion base pairs, right? So we're right around here. And our mutation rates occur very seldom. So on the order of one mutation for every 10 billion, or maybe 100 million replication ends every nucleotide. One error for every 100 billion or 10 billion nucleotides. Very low mutation rates. Even bacteria, if you look at bacteria, they do have higher mutation rates, maybe a lot or two higher mutation rates, and their genomes are considerably smaller, on the order of millions of base pairs. So that's cellular life. If you jump up to viruses now, double-stranded DNA viruses like herpes virus or hepatitis B, very much smaller genomes. The biggest viruses probably still only have genome sizes of around 230,000 base pairs. And these guys have significantly higher mutation rates than all cellular life. Okay. And then from there, if you jump up to RNA viruses, note that you have several orders of magnitude higher mutation rates for these RNA viruses. That again is due to the polymerases that they use. And much smaller genome sizes. So the RNA viruses, usually genomes are around 10,000 nucleotides. SARS-CoV-2 is about 29,000, Ebola is about 19,000 nucleotides, so just for a frame of reference, pretty small genomes. Okay, this is kind of a scary, horrifying chart, but this is what we're looking at now. You don't have to memorize this, but this is the Baltimore classification scheme on how we classify and categorize viruses. We do it according to whether or not they are DNA viruses or RNA viruses. And then from there, the next level down, whether they're double-stranded or single-stranded. And then from there, whether they're enveloped or non-enveloped. This chart overviews a lot of the major clinically relevant human pathogenic viruses that we're going to be talking about in this block. Many of them you'll see are single-stranded RNA viruses, and hopefully many of these you recognize, like Ebola virus, mumps, measles, influenza, Lassa, yellow fever, dengue, West Nile, a lot of really well-known viruses. Those are single-stranded viruses. But that doesn't mean that in the DNA virus world there aren't some significant pathogens that we also need to worry about and think about. We've heard of human papillomavirus before, right? It causes warts. That's a DNA virus, double-stranded DNA virus. Heard of adenovirus maybe not. It's a double-stranded DNA virus. Smallpox, envelope virus, herpes viruses, hepatitis B, some really important pathogens on this list. Okay, so a wide variety, but it's important for you to have this kind of conceptual framework of how they're organized. Going back to the strandedness of positive and negative sense RNA viruses, just to make sure we understand that point. We'll go through a couple of examples of how this works, but I just want to get a warm fuzzy that you guys are following, because historically this does confuse a lot of students, but it kind of directly ties in with our knowledge that we picked up in block 2. I'll try to link that and make sure that we understand what's going on here. So you remember the slide from block 2 when we were talking about eukaryotic, actually it was prokaryotic replication and transcription. But you have this double-stranded template, right? You remember that coding strand is what has the same sequence as the mRNA transcript that comes off. And the template strand is the one that's actually transcribed directly. So this is a complementary sequence to the template strand, but the same sequence to the coding strand. When we talk about viruses, again, that positive sense virus, positive sense strand refers to the coding strand. So if this is all a virus has this packing in its particle, a positive sense RNA strand, this is what first encounters the ribosome out in the cytoplasm and gets directly translated. Does that make sense to everybody? Everybody's tracking on that? Okay. With a negative sense RNA virus, all they have is their negative sense RNA strand, right? So that has to be first transcribed into a positive sense intermediate copy, so that the genes on it can be translated into protein by the ribosome. A couple of examples to show that more clearly. Positive sense RNA virus replication pathway shown on the left. We have negative sense RNA replication process shown on the right. For the positive sense RNA virus, when these particles come in and uncoat and bear their genomes out in the cytoplasm ready to be replicated, they can go straight to that host cell ribosome, and those viral proteins can be translated directly from that. Chief among those viral proteins that get translated immediately is the RDRP, the RNA-dependent RNA polymerase. Once that is translated, that protein will go up and start making the antigenomic copies, and then the genomic copies. So the antigenomic copies again are negative sense, and all the genomic copies are going to be in that positive sense orientation. And those are what gets packaged into the new virus particles. So you have your new viral proteins, you have all your new viral positive sense genomes, and all that gets packaged in huge numbers, right? Hundreds of thousands of these progeny virions getting released. Negative sense RNA viruses, notice in this cartoon they show like a little oval in there. That oval is depicting the fact that they have to bring their own intact RNA-dependent RNA polymerase with them. Otherwise, once they uncoat, that polymerase is not going to start, that ribosome isn't going to start translating protein. So So they bring their own intact enzymatically functional polymerase with them, so you can immediately start transcribing that positive sense intermediate antigenome. And from there, you get, everything else is the same after that point, okay? Everybody tracking on that?
Yeah,
Okay,
I have a question.
Yeah.
Sorry. So the negative sense strand is reflective of the template strand, that's what we said. On the previous
Yeah.
slide.
Yeah.
That's what's,
Analogous to, but if we're talking about RNA, it's just so you can tie it in with.
I understand, but as far as like how it relates to the template, like if we're using an analogy, why would the the polymerase not use that if it's analogous to the template strand, and we use the template strand for DNA polymerase
Mm-hmm.
to make mRNA, why do you have to go and make a positive?
So think about, like, let's just have a 3 nucleotide example. You have an AGA template, and it's your negative sense RNA virus, right? What needs to go into the output virus particles would be an AGA, right? But when you have a polymerase is always going to add a complementary base, right? So you can base pair it correctly. So what RNA base pairs to an A? U, and then G, C, and then the other A. Right, so a UCU would have to be the complementary intermediate. That would be plus sense. And then that's not gonna do you any good for like your actual genome, so you have to make additional copies from that so you can actually be in the right orientation for your progeny. They've evolved to be negative sense RNA viruses. They have their own advantages and disadvantages. But that's, you can't put an A, you can't replicate an A from an A. There has to be base pairing during replication.
Gotcha.
Yeah, does that?
Yeah.
Okay. And that is, I didn't put it in there, but you can kind of play around with that concept and think about it so it makes sense to you, but
And these two are doing the same thing. The biggest difference is one brings its own equipment and the other one doesn't. Correct?
no, they do, just one, so this one is, think about it as being on board RDRP, but it still brings this one still brings its own equipment, it just hasn't been made yet. So it's a gene on that positive sense segment that has to be translated into this protein. But it's still viral origin because the host cell is not going to provide that for the virus. It's got to bring its own.
After that, they're doing the same process that way.
It's essentially the same from there.
So for the positive sense, it kind of only go through transcription one time, while the negative sense goes through twice.
Yeah, we have to be careful about word choice there. But yeah, what's happening here is you have replication of the genome going on here. This is still just regular translation, but here this is replication of the genome into an antigenome. And I think this is where it gets confusing because we don't have this in eukaryotic cells and prokaryotic, like cellular life. We don't have this happening. But transcription from here, there can be late transcription occurring, but what this cartoon is showing is basically how the genome gets copied and amplified. Oh, yeah.
So, I just wanna make sure that I'm wrapping my head around this. So, the negative sense viruses, negative sense RNA viruses, in order for it to continue to replicate and proliferate, the genome has to stay negative sense.
Yeah.
So it has to undergo translation, or transcription and then translation to make more negative sense copies.
Yeah.
In order to proliferate.
Yeah, and think of it this way, if it comes in as a negative sense virus, it's either gonna leave as a negative sense virus or it's not gonna leave at all.
Yeah, it's not really all.
Exactly. If something happens, like let's say the host cell is not able to provide some other factor that it needs to replicate, it will not produce progeny virions. But flu virus, that's a negative sense RNA virus. It's gonna come in as a negative sense RNA virus, it will leave that way as well. So it's not gonna be an infectious competent virion or virus particle unless it packages negative sense copies that is successfully made in that host cell.
But in order for it to make more negative sense copies, it has to undergo transcription with the RDRP.
Yep.
Because you can't make a negative from a negative.
Exactly. exactly, that's a beautifully put way of saying it, can't make a negative from negative. Gotta make a positive intermediate and then more negative copies from that positive intermediate. Yeah, positive intermediate, just remember that term anti-genome, that's what it's called, an anti-genome, but that's a temporary fleeting molecule, doesn't last, doesn't get packaged. Its sole purpose is to make more negative copies of that genome. So, we'll kind of let that, I'll try to ask you some questions next week to make sure that we are tracking on this concept, but let me know if it's still unclear, it usually is to a lot of people, and yeah.
Sorry, Matt, so couple slides back, talking about the properties of the RNA viruses.
Mhm.
For missiles, it kind of go through from negative to positive to negative again. Why does another negative has to be in there?
So let's look at this. So negative to positive to negative, right? Here. So this is a genome that gets packaged, and what this is showing is you can have later transcription occurring from the after replication. So like you have lots of copies of genomes that have been made at that point. You can still have late transcription events. So those would be transcripts coming off of that. Yeah, early transcription, late transcription. Yeah. Okay. Yeah, super confusing topic, I know. In terms of the entire Baltimore classification schema of viruses and what you need to kind of know at this point, the parvoviruses, the positive sense DNA viruses, you don't have to know anything about their replication. You don't have to know that about rotaviruses. These are pretty small in terms of clinical significance. They're not important enough to put much energy or emphasis on how these things replicate. Hepatitis B for group 7, this is a double-stranded DNA virus that goes through reverse transcription. We don't need to emphasize that either. But what you probably need to have a grasp on are just normal DNA, double-stranded DNA viruses like the pox viruses, which is pretty straightforward, but also these single-stranded RNA viruses, because the vast majority of viruses we're going to be talking about in this course are RNA viruses, either positive sense or negative sense. And we won't hit this in great detail except for a few major ones. We'll talk about influenza in some depth. That's a negative sense RNA virus, segmented genome enveloped. We'll talk about COVID in pretty good depth. That's a positive sense RNA virus enveloped. We'll talk about HIV and hepatitis B, but other than that, we won't get into replication much. And the reason why we bother to do this, beyond the fact that it's just like basic virology, is you don't really have a good understanding of the few specific antiviral meds we do have until or unless you have a good understanding of how viruses replicate. Okay, so that's kind of the main justification there. And also with mutation rates, just kind of keeping up with the meaning and significance of particularly RNA viruses that mutate all the time. HIV, lots of mutations. COVID, lots of mutations. Influenza, lots of mutations. You kind of need to have a sense of how viruses replicate in order to appreciate that. Okay, so with that, we will go into these steps, which I think we covered a little bit, right, in block 1 during the intro, how viruses replicate. So, in terms of the text, we can kind of blast through this, but I'll spend more time on figures. I like us to sort of visually follow what's going on with this infectious cycle of viruses. But in essence, you start off with an infectious virus particle, a fully competent infectious virus particle, and a susceptible host cell. The virus will come along and it needs to get into that host cell, right? So most viruses have a specific tropism for specific cells. We used that word tropism earlier. What does tropism mean? Yeah, an affinity or propensity to infect something specific in this context. So they'll have their tropism, and that early tropism is conferred by the type of glycoprotein they carry on their surface. So they have a glycoprotein. Usually those glycoproteins will only bind to certain host cell receptors, and that's sort of like the initial handshake that this is going to be a viable cell to infect. Once that initial interaction is made, oftentimes, depending on the virus, it will trigger receptor-mediated endocytosis. That entire virus particle gets endocytosed into that cell in an enveloped compartment called an endosome. If If it were to stay in that endosome without getting out, it would probably be destroyed because those endosomes eventually start acidifying. The pH drops and they become super acidic. But rather than getting destroyed, most virus particles will use that low pH, the dropping of the pH, to trigger conformational changes that allow them to get out of that endosome and into the cytoplasm where they need to be. Okay, so once they get into the cytoplasm, their envelope is gone. Let me skip it. Well, this is not a super great figure from that, but their envelope is gone and they have their encapsulated genome at that point. They have to get uncoded in order for their genome to be replicated. So they'll uncode, they'll lose their nucleocapsid covering, and then they will use either host cell machinery if they're DNA viruses or their own polymerases if they're RNA viruses to start replicating their genomes and transcribing those viral genes. Once all of those things happen, the transcription, translation, and genome replication, like the major synthetic steps, then the viruses need to assemble, self-assemble, and then they need to get out of the host cell. So that's called release. With naked viruses, a slightly different process again. Remember we said they kind of build up in numbers. These viral progeny viruses will build up in numbers and then burst out of the cell in lysis. RNA or envelope viruses, that process is a little bit differently. Individually bud out of the membrane in a more gentle manner that it can be survived by the host cell in small numbers. So this is injurious to the cell, the release step. In and of itself, even envelope viruses in great numbers will hurt that host cell. But what you need to kind of keep in mind is it's the synthesis steps that are most taxing on the host cell. The host cell has to shut down all of its normal translation and transcription machinery and basically divert those resources to supporting viral replication. So a muscle cell that's infected stops being a muscle cell because it has to serve the virus. Or a respiratory epithelial cell stops doing what it needs to do. A neuron stops doing what it needs to do. So not only does it lose its functionality, but over time it can completely destroy the cell even if it doesn't replicate by lysis, just by sheer exhaustion and depletion of resources. Either way, the outcome for most of these acute lytic viruses is the cell ends up getting destroyed, despite whether it's an envelope or a naked virus. In terms of what this looks like with this diagram, I do kind of hate this diagram, but it's nice because what it's showing is the difference between RNA viruses and DNA viruses. This is a contrived cartoon, it doesn't really happen this way, but let's say you have an RNA virus entering a cell. The main point here is what we've already said a couple times. The RNA viruses will replicate in the cytoplasm, right? The DNA virus you need to pay attention to and know here is that a DNA virus needs to get into the nucleus in order to get replicated and transcribed by host cell polymerases. There are some exceptions to this, we'll talk about some of those exceptions, but by and large, that's the difference between the RNA viruses and the DNA viruses with regard to where in the cell they're replicating. And even for these DNA viruses, once they've transcribed viral mRNA transcripts, just like host cell transcripts, they need to be exported out of the nucleus because that's where the ribosomes are. Okay, so then they get translated into proteins in the cytoplasm. All right, now putting this together a little bit more, we have a timeline that is good to know, so you know what, when we talk about incubation periods, you have an understanding of what's going into these incubation periods, what's happening. Susceptible host first gets exposed to a virus, they might not get sick for a couple days later, but what's happening during that incubation period are all these synthetic events. That takes time, from the time that the virus first encounters a susceptible host cell to the time that you make progeny virions will take anywhere between 1 to 20 hours depending on the virus. So it's a huge variability. Some viruses are much faster. Phages, the bacteriophages that we talked about that will target bacteria, those are really fast. So super short synthetic pathways. But up to 20 hours. After that time, you have your progeny virions that are being released out to the host to go and infect other cells. And depending on the virus, you could have thousands, like tens of thousands or hundreds of thousands of progeny virions being released during a single burst. If you do the math on the number of susceptible host cells available for these infections, intuitively, you do understand that you're gonna run out of susceptible cells pretty fast, right? You can't really sustain that exponential growth. So you do have decay of those viral progeny pretty quickly. Many of them are lost to the immune system. Many of the infected cells are destroyed by apoptosis, which is an important immune function to kill infected cells by apoptosis. Some of them just deplete the resources of the cell. So at any rate, the amplification does not continue exponentially. There are things that control the population of new virus particles that are being created. But regardless, you still have this period where you have exponential increase before you have things that kind of get that under control, and that is this exponential phase. This, again, highly depends on the type of virus, but usually between 8 to 72 hours is when you start to see high levels of viremia from these replication events, new viruses that are being created. And this can kind of start to dovetail with where you see symptoms in acute infections. We're talking about a common cold, when that person might start to feel sick at that point. But it's not going to be during the eclipse period, unless there's some like immunopathology involved. Okay, in terms of viral infections, we'll go through this slide and take a break after this. We have a couple different types of viral infections. The ones that we usually think about are the acute infections with lytic viruses. So in this example, the host cell is almost invariably killed. You make a bunch of progeny viruses. And then the host, as an organism, if we're talking about a human being, there's usually two outcomes when you have a lytic viral infection. Either you clear the virus or you die as an organism. Let's say it's like Ebola virus, right? Ebola can kill you, it's an acute infection. That's one outcome. CFRs for Ebola infections can be up to 80, 90%, right? So it's an extremely lethal outcome for the organism. Most of the acute infections that we're going to talk about in this course are not fatal outcomes. If you're talking about like paramyxoviruses, little common cold viruses, things like that, the immune system will eventually get the upper hand, and usually within a week or two. Okay, so most of those lytic virus outcomes are that you end up on top of that, but they can be also fatal. Another category are abortive infections. This is when the virus tries to infect a cell and is not able to for whatever reason. The host cell doesn't have the right enzymes, it doesn't have the right whatever other features are relied upon, but you don't get productive infection. So no progeny viruses are produced, and you can get like a garbage pile of virus particles that maybe they won't assemble, some things like that. So limited pathology with abortive infections. I'm sorry for the weird animation on these slides. Get rid of that. Third category are persistent infections. These are, again, more associated with DNA viruses. They get into the cell. These DNA viruses oftentimes are able to survive for very long periods of time because they make super low numbers of progeny virions. So it's not very taxing on the host cell. The host cells can survive, or if they do get killed, they get killed over time. They may be able to have limited capacity to even have their own normal functions, so their normal cellular functions. Examples of those persistent viruses are things like Hep B, Hep C, and HIV. Last category are latent infections. A virus that establishes a latent infection can persist for decades, and that's because no virus particles are being produced. With these, there's minimal activity. They're basically inert in a host cell, oftentimes integrated into the genome. And that wouldn't be a problem if it weren't for the fact that they can be reactivated down the road. So when they get reactivated, that means they start transcribing viral genes and translating viral proteins and copying their, so they basically just come alive again, if you want to think about it that way, become productive infections. So latent infections would kick back up to becoming lytic infections. Herpes is a famous example of a latent virus. Okay, let's go ahead and take a 10-minute break. I've got 10:50 starting up at 11.
Question for you. I know some of us are still tender about this whole eclipse period.
Yeah.
On the first test, there was a question, and all like, I don't know how many people, were like, yep, eclipse phase, but it wasn't the eclipse phase. But it was the period, I swear the question was like the period between initial attachment and host cell assembly in the first batch. I don't know if you remember that question, but like, a lot of us got it wrong, because we put that it was the period between the initial attachment of the host cell and assembly of the first eukaryotes, but it's like a different. Yeah. I just know that was a big one. Everyone was like, eclipse period. And so,
I mean, I can look at it, I don't know that though, I'm extremely interested in
exactly. Maybe it was because it's worded a little bit, so a lot of us saw the question and we're like, oh, eclipse period. I don't know if it said that what is the eclipse period, or if it gave us the definition of what period is this.
Yeah.
And it wasn't, it was like an maturation of the and it was like all of it. What?
Wow, I promise you I will solve that question tonight. I'll bring it up,
But yeah, maybe it's maybe the questions were different, but I know that was a big point of like,
yeah, interesting.
I kind of told myself I walked in today.
Basically kind of just showing differences in but it ties in with
I think the micro exam imaging is kind of
whether or not it is like it comes into the cell.
we'll see how
It's not ready to be translated. It's not ready. Exactly. So it's hard to work in counter. So we're able to read so it's not right orientation. Yeah, this is why it's like,
this I read it, I'm like, oh, I see. I read it on the slide first, I just wrote like, yeah, I wanted to do this. And then they have to bring their own functional or this weekend.
yeah, okay. Yeah, okay, that makes sense. Yeah, and then when that happens, it's very stuff, it's the same as the
If it comes in negative, it has to be negative.
But
That's like at least the rough. Positive just comes in and it immediately jumps into the cells representative of negative. It just copies the cell. Positive make it negative. Yeah. What it's all the positive is still cell ribosomal, but it also can make its own RCD, which can then go over and replicate itself. So it can go over it.
because it was, we were trying to, what was your question again? The last, why do we need to do it?
Why do you have to make it, right, to make it right? That's what you're saying, it's like,
Why don't we just use that?
why do I gotta make that, and now that's, now I can make that.
It's like, okay, first two.
We say like, oh, negative three is like.
It's stuff like that. Ever since I was talking about my detail and all this stuff. Things are because they are. And because so many people have said that they are, it's way it is. Honestly, that's hard to are we sitting there watching every step of this process? We infer it, and now it's documented. It's 100%. Like, because everybody accepts it. It is not everything. It's the way. What if it doesn't? Oh, you didn't jump. You know, but people have to say true metal. Well, you won't fast. Dude, I completely don't believe
to start back up again. We're done with intramaterial, ready to start getting into viral pathogens now. Like I said, we've got intramaterial, I will develop some questions for next week, but do let me know, a couple of you asked me some questions just to confirm what's going on with that process, but I never do a great job of explaining it, and it is super confusing. But we'll make sure we get there. Alright, and maybe some of that will become a little bit more apparent too as we start talking about influenza, which will be the first thing on our agenda. We'll talk about the way Block 4 is organized roughly is by, sort of by organ system and that's affected. So we'll talk about these respiratory viruses first, and then we'll talk about herpes virus, a couple GI viruses that are important, HIV, hepatitis viruses, the hemorrhagic fever viruses, rabies, and then some skin viruses. That's roughly how we're gonna go through Block 4. So, starting with the respiratory viruses, these represent different viral classification groups. Influenza is in its own group, Orthomyxoviridae, again, negative sense RNA envelope virus. Rhinovirus and enterovirus are capsid viruses. They're DNA viruses. They actually are related to poliovirus. So rhinovirus we'll talk about a little bit because it's important. It's a common cold virus, but enterovirus D68 we won't talk about until we talk about polio later on, even though it too can cause respiratory symptoms. Coronavirus, we all are familiar with coronavirus. That's a positive sense enveloped virus, positive sense RNA virus. And then RSV, parainfluenza viruses, and metapneumovirus, those are all negative sense RNA viruses that are enveloped. And these three middle ones are pretty related to each other. Everything on this list is mostly associated with common cold viruses, but influenza is sort of like its own thing. So always important to know about influenza, and that's why we're going to study it in more detail. And in doing so, hopefully some of these other concepts that relate broadly to virology will settle in. We'll start off with structure, morphology of influenza virus particles, kind of working our way from the outside in. Envelope virus, so the exterior is derived from host cell plasma membrane, lipid bilayer right there. But we also have some viral protein, glycoproteins that are on that membrane. Two of them are very, very important. Hemagglutinin, which is sometimes abbreviated as HA, or sometimes just H, and then neuraminidase, which is sometimes abbreviated as NA or N. Neuraminidase has enzymatic activity, notice that suffix ASE. What it does is cleave sialic acid residues at the tail end of the infectious cycle when those viruses bud from, or starting to bud from the host cells. They would remain tethered to sialic acid were it not for the enzymatic activity of neuraminidase. So it's very important there. And then hemagglutinin is really important because that is the viral glycoprotein that interacts with the host cell receptor on susceptible cells. Anybody know what the host cell receptor is for influenza virus? What it's binding to? Okay, sialic acid. We'll see that in a couple slides, but hemagglutinin is the one that binds to that. It's really, really important. We'll come to appreciate why that's important more in later slides. Moving inwards from the exterior, we have a matrix protein that lines the interior of the virus particle. It gives its shape. This is called M1, matrix protein. It's the most abundant viral protein, and it's oftentimes what we could look for if we're doing Western blots and things like that because it's so abundant in these virus particles. The M2 protein is also on the membrane. You can see that on this cartoon here. It has an important role during uncoating when these viruses are in the endosomes when they've been endocytosed. And they allow for proteins to enter this virus particle and allow for uncoating to happen. So this is important to know because it's a target, a drug target for anti-influenza drugs like phenantidine and amantadine, those anti-influenza drugs. Nucleocapsid protein, you can see NP, that is the capsid that is covering the genome, the RNA genome. So that helical capsid and a close association with the genome itself. And with this genome, real quick, notice that it's on 8 different segments. These segments encode each at least one viral protein. This is really unusual for viruses. Most viruses have unsegmented or single segment genomes. This has 8. So that has important implications as well. Then we have a nuclear export protein that allows this virus to get out of the nucleus. It's one of the only exceptions to what I was just explaining with RNA viruses being in the cytoplasm. Influenza happens to require being in the nucleus briefly because its transcripts need a 5' cap. Most RNA viruses can supply their own, but this one cap snatches or steals from the host cell. So it does need to go into the nucleus to grab a 5' cap. Don't worry about that too much. It also has a tripartite RNA polymerase, RDRP, that's PB2, PB1, and PA. So there are at least 8 different RNA polymerases that are built into these virus particles. When uncoding happens, these polymerases start getting to work, making new copies of that genome, synthesizing new copies. And then finally, we have PD1F2. This is not part of the polymerase, but it does function in dampening down some host cell responses. So it'll help prevent that cell from committing suicide as an anti-apoptosis function and things like that. So those are the basic important features of the structure and function of influenza proteins. In terms of replication, we can kind of study this replication cycle with the lens of which drugs have been developed against influenza virus. A lot of viruses we don't have specific antivirals against, but influenza is one that we've developed a ton of different specific antivirals, and they target different aspects of the infectious cycle. We don't have to go through all of these, but there are a couple that are good to know. Let's just talk about how this virus infects the cell, and hopefully we'll kind of reinforce that cycle a little bit. So you have your intact virus particle. It's coming into contact with a susceptible respiratory epithelial cell that expresses this receptor, sialic acid, on the exterior. When it binds to sialic acid residues, it gets endositosed. Once it gets endositosed, the dropping pH in that endosome will trigger that virus to undergo uncoating, and the lipid exterior, that lipid envelope exterior, will fuse and merge with the endosomal lipid by there. So once you have that fusion, you can see as we progress through this cartoon, the genome segments, these little wavy type patterns right there, can be dumped out of that particle, and they're basically just covered with a nucleocapsid at that point. That nucleocapsid does need to go before polymerases can go do their thing and start replicating antigenomes to genomes. As those, as positive sense transcripts are being synthesized from this native sense native sense RNA virus, they will start being translated into protein out in the cytoplasm where the ribosomes are. And then those proteins and new viral progeny genomes will start assembling. You get a little marker going so we can kind of see the whole class stuff and toggling back and forth. So at this point we have new proteins, we have new genomes, and assembly is happening at the plasma membrane. Viral glycoproteins are being inserted here. Host cell proteins are being displaced. So at the site of budding, what ends up budding out of this is a lipid envelope that's enriched with viral glycoproteins, and it has a whole intact viral core at the center of it. Because of the sialic acid and hemagglutinin bond that happens, those new particles right there that are trying to bud off will stay tethered, and that again is why you need neuraminidase, the viral neuraminidase. That neuraminidase will cleave that bond, and it'll allow that infectious particle to get released off into the milieu and infect new cells. Yeah. No, and not every cell has the right linkage of sialic acid as well. So this has a specific linkage, it's called a 2,6 sialic acid linkage, and it is prevalent in the respiratory tract. It makes those cells susceptible to influenza. That's why you don't usually get like influenza infections in your kidneys, for example. It's different linkage, different cell receptors.
I thought the I thought like a big distincting factor was between RNA and DNA is that RNA is in the cytoplasm, is in the cytoplasm, and it lyses out. But this one is going in the nucleus and budding off. So why not just consider it a DNA virus?
Well, it is RNA, so it did have, its genome itself is RNA. But it is true that most RNA viruses only need to be in the cytoplasm because they have their own specific polymerases. This has its own specific polymerase, but the reason why it's the exception to that rule about RNA viruses is because once it has its positive sense transcripts, they don't have a 5' cap. And they need to get a 5' cap from what's going with them in the nucleus. That's the only reason why this one type of virus is the exception, this one type of RNA virus. But most RNA viruses will just stay out in the cytoplasm. That's the only reason. In terms of lysing, this is an envelope virus, so when it is ready, like when we're at this point right here, it has its capsid, it has this nucleocapsid protecting its genome, but it's just going to press up against the plasma membrane and be encapsulated by the host cell envelope. Okay, so there's no lysis going on. This is like that gentle budding effect. And if you look at this, you guys see this electron micrograph at the top there. That's showing this gentle budding off the virus particles. Which again, if you have one, not a big deal, but you have hundreds, sometimes thousands happening, and you're losing a lot of membrane at that point. So that will eventually destroy the cell, but it's not in a spectacular lysis fashion that you would expect with like a non-envelope capsid virus. Does that make sense?
Yes.
Okay, perfect. Okay, so druggable targets here. M2, that protein that allows for uncoding to happen, this was a primary target when they developed drugs like amantadine and rimantadine. Unfortunately, these are pretty much no longer used therapeutically because of resistance to them, antiviral resistance to amantadine and rimantadine. So primarily, even though a lot of these drugs have viable targets, the one that you need to pay attention to is right here. Neuraminidase inhibitors is a class of antivirals for influenza. So these would be drugs like oseltamivir, Tamiflu, if you've heard about that. We don't have significant resistance to oseltamivir yet, but we use it judiciously as well. But that's probably the frontline drug right now for influenza, moderate to severe influenza cases. And what that drug does basically is it prevents neuraminidase from cleaving the virus particle from sialic acid on the surface. Okay, talking a little bit about epidemiology and the types of different influenza viruses. As I've mentioned, when we're talking about influenza, that's sort of at the family level, the Orthomyxoviridae, segmented into negative sense RNA viruses. But we do have the equivalent of genera that fall underneath this. Influenza A as a genus, influenza B, C, and D. A and B are the ones that are really important to know, that we do active surveillance for, that we build our vaccines around. So super important. We'll spend most of our time talking about influenza A and influenza B. I've color-coded the text so that it matches and corresponds to this phylogenetic tree that shows the relatedness of these different influenza genera. But like influenza A, for example, is in black here. And the point of looking at influenza A is to appreciate the fact that influenza A has a huge number of animal reservoirs, and they're kind of grouped together by relatedness. But you see like a beluga whale silhouette on this thing. It's because, you know, whales are mammals and they can get flu as well. So we get a huge variety of different animals can get influenza infections. The main reservoir, though, are birds, avian species, in particular aquatic avian species like ducks and geese and swans. Although domesticated poultry populations as well can carry like a be appreciable reservoirs for influenza. In terms of how this is broken down, with influenza A, we have subtypes of the hemagglutinin and neuraminidase we were talking about, and that confers which type of subtype of influenza A we're talking about. We have 16 different types of hemagglutinin and 9 different types of neuraminidase. Lots of different combinations are possible, and with those combinations, we have different animal species that are associated with carriage of those. With the human adapted species or types, subtypes of influenza, we have predominantly, and this is why I like this figure so much, it actually shows you which subtypes of hemagglutinin are associated with human populations, like human adapted. You've heard of H3N2, you've heard of maybe H5N1, maybe H1N1. When I say those H's, that corresponds to a human adapted subtype. So this H2 is human adapted, this H1 is human adapted. This H3 is human adapted. So on and so forth, right? When you look at H5 down here, I want to point that out as well. H5, yeah, so what are the birds, what are the species silhouettes that looks like it's pointing to? Yeah, domesticated chickens perhaps, and some sort of wild bird. So that's an avian influenza subtype, H5. About a year ago, do you guys remember what was in the news a lot about a year ago as it relates to avian influenza? Something about chickens. Yeah, chicken pox. Yeah, that's sad. No, it was like H5N1. Does that ring a bell with anyone? This time last year, maybe a little earlier, April 2024. We were worried about H5N1. And we were worried because we were starting to see cases occur in livestock, this bird flu, which was suggesting that it's making a species jump from cows to people. You know, we were doing a lot of surveillance on that. And then we started seeing cases happening in people. So we had about 70, maybe 76 cases of H5N1 in people. We had a couple US states that had H5N1. And we were worried that this virus was going to start adapting and being transmissible person to person, which takes, it's not likely that that happens, but if it does happen, we worry about that a lot. Fast forward a year, here we are in July of 2025, we've just stopped surveillance of H5N1 because we're not seeing a lot of evidence that it's being transmitted. So it's controversial, a lot of people are freaking out that we're stopping surveillance programs right now. Surveillance programs on like dairy farms and things like that, but it's because we weren't seeing transmission. But a year ago, that was a big concern. At any rate, these are the human adapted subtypes when we're talking about influenza A. The reason why we care so much about influenza A is because these are the the genus that's associated with pandemics and it has an unstable genome. We'll talk about that in a little bit more detail too. Yes, sir.
Spanish flu type of.
It was, yeah, influenza A, absolutely. H1N1, Spanish influenza in 1818. Yeah. Yeah. Influenza B, so in red here, what do we see about the representative species diversity of influenza A or B? Seals, yeah, oddly enough. So, yeah, seals and humans, but a much less diverse mammal reservoir population. And because of that, there are some implications for how it circulates in humans. We don't have subtypes, so there's no such thing as like an H3N2 influenza B. It has a relatively stable genome. In the human population, we do surveillance for it. It can cause significant disease, viral pneumonia, influenza. It generally has more like a year-round slow burn number of cases, although there's a little bit of seasonality to it. So yeah, limited pandemic potential, stable genome, and we don't have reassortment, which hopefully is ringing a bell. Talking about this in block 2, we'll talk about reassortment more as well. Disproportionately affects children and only minor antigenic changes. So this antigenic drift is something else we'll talk about in a slide or two. And then lastly, color-coded here, blue, C and D. We do get cases of influenza C in the population, but these are generally pretty mild. They would be more like a common cold presentation than what we would consider a flu presentation with influenza A or B. And there is an influenza D, but that does not infect humans. That is restricted to cattle and swine populations. All right, so antigenic drift and antigenic shift. You may have heard of these terms before. There's a big difference between them. Starting with the more subtle one first, antigenic drift, and what that means, what the implications are. This just refers to the normal mutations and errors that occur with every round of replication, as these viruses are replicating their genomes, they make errors. We saw on that chart, there's a predictable number of mutations that occur with every round of replication, and those errors over time accumulate and might have changes and effects on things like transmissibility and how the viruses bind to host cells and things like that. But that is a slow evolutionary phenomenon over time. Antigenic shift occurs with influenza A viruses when they resort in host cells. So the conditions under which that happens are you have to have not super closely related influenza A viruses that co-infect a susceptible cell at the same time and undergo reassortment. Reassortment, you guys remember, what does that mean in terms of expanding genetic diversity?
You have two different viruses that ended with the same cell at the same time. They swap DNA, you get a whole
It's about RNA segments, but yeah, exactly. So because of the fact that these guys are segmented, they have 8 segments from one virus, 8 segments from another virus, so you have 16 segments. The virus doesn't have a way of packaging them as they came in, so you get reassortment of different segments together. Because of that, you get entirely new subtypes that get into circulation. And if those subtypes are capable of undergoing lytic infection without being abortive in a human, then you can get pandemics. This is how pandemics of influenza happen. They usually happen 10 to 20 years, sometimes less frequently than that, but the 20th century, we had some doozies, right? We had some big influenza pandemics. Starting with 1918, like I said, that was an H1N1, an avian influenza virus that was responsible for anywhere between 50 to 100 million deaths worldwide. Extremely virulent. People didn't have immunity to it. They had never seen it before, so huge, kind of never before seen consequences for that. 1918. 1957, there was another big influenza pandemic. It was the Asian flu. Much less virulent, far fewer people died during that pandemic, but it still put into circulation another type of virus. That was an H2N2 influenza A virus that caused the 1957 pandemic. And then just a short time later, 11 years later, we had the Hong Kong flu. That was an H3N2 influenza A virus. 1977, reemergence of H1N1, and then in 2009, another big seminal event. This was all within probably a living memory for everybody in this room. You guys remember the 2009 swine flu epidemic, pandemic? A little bit. Yeah, so that replaced it became the dominant circulating pandemic strain of influenza A at that point in time. And we still have H1N1 swine flu circulating today. But that was, at least compared to those other pandemics, relatively milder than the previous ones. It had about 150,000 to 575,000 cases worldwide. All right, just a couple more slides to make sure we understand the implications here. This is showing the conditions under which reassortment can occur, and does oftentimes occur. What you have is an intermediate species like swan that tend to be susceptible to influenza, so they make good mixing animals for reassortment events. What this cartoon is showing you is a susceptible swine. It comes into close contact with a human being that has a human adapted flu strain, and then some domesticated poultry animal that has a different avian adopted flu strain, comes together, reassorts, reshuffles, and then you get an entirely new virus out of that that may be able to infect, let's say, domesticated poultry, or might be able to immediately infect human populations. When this antigenic shift happens, again, we don't have immunity to it, something usually that our vaccines, our annual flu vaccines haven't incorporated. So most of the population, when we say they're immunologically naive, it means that they've never seen that before and they tend to be super vulnerable to those infections. Yeah, John.
With reabsorbing them, is it possible to have a virus be both a positive and a negative sense?
No, no. So if you were to like accidentally package a positive sense intermediate strand, that, I don't know if that could even happen, but that would not be viable. At the end, there's probably a lot of mistakes and weird aberrant things that happen during these synthetic events, but at the end, what the virus particle needs to be in order to be infectious is to have a viable genomic architecture. And for influenza, that's 8 negative sense RNA strands with the 8 requisite that encode for those viral proteins that it absolutely requires. So when we start getting these emerging or adaptive avian subtypes that we're worried about that aren't quite human adapted yet, things like H5N1, which is what I was talking about previously, and H7N9, that's another one that we're worried about, an avian influenza strain. We monitor those very carefully and closely with the concern, again, that those zoonotic influenza viruses will become adapted to humans and be able to spread efficiently from human to human. Zoonotic spillover cases from interacting with, a cow at a livestock show or a chicken or something like that, one-offs, those are bad, but they're not concerning because, again, the major thing you're worried about is adaptation from human to human transmission, not animal to human. When we do identify, they're called HPAIs, highly pathogenic avian influenza, that's when we take pretty drastic measures, not just we as Americans, but worldwide. Countries will take drastic measures and you start these like bird holocausts, right? Where they start pulling all these animals. One of the reasons why egg prices occasionally go up is because you have culling of poultry populations. But the figure on the right here, that's what this is showing, a bulldozer, and its load is a bunch of dead chickens that probably have influenza circulating among them. And on the left, what you see is an infected chicken. So they do, like, when chickens are infected with influenza, they do show signs and symptoms. They get sick, they get depressed, they start, like their feathers start ruffling, and their behavior is different. And then you test and do surveillance for them and wipe out an entire colony? I don't know if that's flock, thank you. I was gonna say herd, I was like, that's not right. Yeah.
The reason we get most concerned about avian influenza is because of the potential for spread because of actual like migratory birds? Or is it like something about bird cells or like respiratory cells that are more analogous to worms? So the evolutionary means
Yeah.
a lot shorter.
Yeah, so it's kind of a complex ecology. Migratory birds do play a significant role in spreading across wide areas, potentially highly pathogenic influenza strains. So that's an important role. In terms of their susceptibility as a host, they have sialic acid residues as well, which makes them susceptible to infection, but they have a different linkage. So part of the challenge that the virus has to overcome in order to make that leap from an avian reservoir to a human population is it has to be able to adapt to using a different sialic acid residue receptor and some other factors as well.
Is theirs like more challenging to overcome than ours? So the concern is if it can make it over that, then it's basically a step down in terms of
Maybe, and it is a little bit of an oversimplification to put all of that spotlight solely on the receptor. There's an entire immune system difference too to contend with, and other physiological differences, body temperature, habit, things like that. But it is considered to be a rare event for a virus to be able to adapt from one species to another. Fortunately, if it were more common and easier for them to do, we would probably not exist as a population, we'd be wiped out. So it's rare and it's difficult for a virus to adapt from birds to swine or from birds to humans or whatever species, but it does happen and it has to overcome a bunch of different cellular and organism level differences that happen. This was a hidden slide from last year. I will not go over it. You should have it as a hidden slide. It's not testable. I just put it in there because it's of interest when we were freaking out about this a year ago, what was happening. doing surveillance in these different states, CDC was putting money and funding into surveillance of dairy farms, like I mentioned. We were trying to make sure that we were detecting any cases. And that is what I was mentioning, the funding has just been more or less stopped for those surveillance efforts for H5N1. Talking about that, we'll stop talking about epidemiology here, this is the last slide, but just real quick to illustrate how unusual this is. This picture, this is from the Murray textbook, but what it's showing is all the very certain events that have to happen in order for that 2009 swine flu to exist. You had to, it was considered to be a triple assortment event, but really it was more of a quadruple assortment event, extremely unlikely. But what happened is you had an avian influenza, a human influenza, an H3N2, and a swine influenza, all co-infect a single susceptible swine species. So now you have a swine that has a reassortment virus in it. If you look at these different segments that are labeled according to avian, human, or S, or swine, A, H, or S. And then that swine mixed in with a couple other swines that were also infected with influenza, different strains. One of those was a Eurasian swine, H1N1, H3N2. And those in turn recombined to another pig, and now you have a quadruple reassortment with Eurasian and non-Eurasian swine, avian, and human adapted segments. So there's a lot going on here, there's a ton going on here. And that virus that was adapted to this particular swine came into close contact with some susceptible human being, and as a result, that human being got sick with this quadruple reassortment influenza A subtype. This was out in California, and then they proceeded to cause a global pandemic because it was adapted for human-to-human transmission. Extremely unlikely, but that's what happened with the 2009 pandemic. In terms of the nomenclature here, it's the A in California for 2009 strain. Influenza A identified in California. 4 was just the isolate designation given to it. And then 2009 is the year that it was identified. So, and then it happened out of season too. This happened, I think, in like early spring, which is out of season for normal influenza season. Usually a good indication that you have something weird and unlikely happening. In terms of the pathology now, just switching gears away from epidemiology, you inhale these viruses that are carried by respiratory droplets. They can also infect you through your eyes if you're not aware of the fact that you can have transmission of viruses through the tears that feed into your nasolacrimal ducts and then down into your nasal cavity and into your pharynx. That is a route of transmission that's underappreciated for viruses. But respiratory pathways and or eyes, generally the eyes, virus enters, it starts replicating locally. This figure on the right, which is also from the Murray textbook, kind of shows you some of the major pathology events that happen. In the green here, we have initial inoculation, some replication in those respiratory cells that have sialic acid on the surface of them. And then, because those cells are dying over time, you have some effects of pathology directly related to the virus itself, death of those respiratory epithelial cells. It will cause desquamation of mucus-secreting goblet cells. Goblet cells in your respiratory tract as well. That's only part of the story that feeds into what we know of as influenza syndrome, though. So direct viral pathology, but we also have a major immunopathology arm to that disease as well. What happens there is once you start have replicating virus, you have your adaptive immune response kicks in again, and just like we talked about yesterday, with these intracellular pathogens, CD8 T cells will get involved, will start killing those infected cells, trying to curb the problem, kill off those cells, cytokine release, interferon gamma again. So some of these signs and symptoms that feed into influenza are also from our own immune system. That's a considerable part of it. Most of the time you just get influenza, which we'll talk about in detail what the symptoms are. But occasionally you get more serious secondary complications like bacterial pneumonia, which going back to the 1918 flu pandemic, a lot of people died of secondary bacterial pneumonia, not the virus itself, but the fact their lungs were so beat up by influenza that they got secondary bacterial pneumonia and died from that. So that's possible. You can progress to primary viral pneumonia, where your lungs are really, they lose their oxygen carrying capacity. You get ARDS, acute respiratory distress syndrome, die from that. And you can even occasionally get CNS involvement. You can detect influenza in CSF and it can cause aseptic meningitis. We don't usually think about that, influenza and aseptic meningitis, but that is a rare and unusual but possible complication for influenza as well. In terms of epidemiology, we have seasonal peaks, famous flu season. We are kind of as far away from being in the flu season as you can imagine right now in summer. It's at its nadir. There's hardly any flu cases happening. But right around the corresponding time of the start of FY26, that's when flu season kicks off. We'll see a big jump in cases right around that time. We'll see influenza A. And then those slowly start to taper off until around May. Looking at some CDC data, the only reason why I'm showing this is to kind of encourage you guys throughout your careers to stay on top of what's circulating. You have to know what's kind of circulating and know where to go to find this data. But CDC's FluView is a really good resource to monitor what's circulating that year because it does change a lot. Can you guys see on your slides when you're looking at this tiny little bar graph up here what the legend corresponds to, the color coding for now? Okay, so this is what we're looking at here is flu season from 2017 to 2018, starting at week 40, which corresponds to about October, and then going to May where it ends and the rest of the year. What do you guys see as predominant circulating influenza strains during that season? Yeah, so H3 influenza A is predominant. In terms of influenza B, we've got two lineages there, Yamagata and Victoria. Yamagata is a light green one. Which of those two do you see predominant for influenza B? The light one, Yamagata predominantly, but far less so than influenza A, right? All right, so that's one flu season. Let's skip ahead a year, and now we're in 2018 to 2019. There's a couple things different. One, the flu season just right-shifted a little bit, but more importantly, what do we see in terms of the distribution of flu strains circulating the following year? Yeah, a huge shift. Yeah, and hardly any flu B. Always pay attention to on the y-axis for scale, but scale hasn't dramatically changed. We're still talking about thousands, in the range of thousands here. So big shift, far less flu B, lots of flu A, and specifically now H1N1, that pandemic strain that we were talking about. Another year forward, now what do you see? B is back in a big way, yeah. And then again, slightly different timing of the flu season, abrupt curtailment there. Now let's look at what's going on this year. What time period are we looking at now? Yeah, we're in COVID years. So look at the y-axis here. How many cases of influenza are we capturing during COVID? Absolute garbage, right? Statistically not meaningful, frustrating. I can't imagine what it was like to work for CDC and flu strains at that time. But yeah, so it's screwed over data. The following year, we're in 21, 22, still very much COVID time, right? So, this data is undoubtedly impacted by COVID, but at least the numbers are starting to return. And now we see a return of a dominant type of influenza, which is what kind? A, yep, H3. Hardly any flu to need in. One more year, we've got 22 to 23, things are starting to normalize. It's looking like a normal flu season finally. And again, we see H3, influenza A, H3. And then lastly, this is from 23 to 24. Pretty nice normal flu season, some cases extending out past May into summer months, but a preponderance of H1N1. Okay. The point of all of that is to kind of illustrate the difficulty in making your annual flu vaccine. If it's shifting so much, the influenza A strains are shifting from H3N2 to H1N1, back and forth, that requires a lot of resourcing to do surveillance and capture whatever's circulating from the year before, and then model that into predictions of what's going to be circulating the next flu season. We've already formulated our flu vaccine for the upcoming flu season. It should be getting released here soon. But we'll see how it does. What I have here is the 2024-2025 last season's flu vaccine and what it had in it. For influenza B, we took out the Yamagata strain. It had just basically gone out of surveillance, as we saw. Replaced it with Victoria, the Victoria lineage. And then for influenza A, we had two different subtypes incorporated, H1N1 and H3N2. Also noticed that we had different places associated with those. One was circulating around Thailand and the other probably in Australia, in Victoria, Australia. It's an egg-based formulation. There's also a cell-based formulation. It's slightly tweaked, but it still has H1N1 and H3N2. I know, crazy. Yeah, Wisconsin and Massachusetts. So, yeah, that just gives you like a little bit of flavor for when you start seeing patients who have influenza, and about a quarter of all visits do relate to respiratory complaints. You can have a little bit of background information about the trends in influenza. Okay, a couple more slides and we'll call it a day, but it is good to make some progress with this deck and get into it. In terms of diagnostics, these are pretty common diagnostic methods. You're probably familiar with the fact that we do have a rapid antigen test for influenza A. And that rapid antigen test will tell you whether it's influenza A or influenza B. So those are commonly used. They don't have super high sensitivity, but they're good enough. They're detecting antigen in the nasopharyngeal cavity. So if somebody is at the very beginning of an infection or the very end of an infection, there might not be a ton of antigen. You might get a false negative, but generally speaking, those are sensitive enough to use if somebody is symptomatic and needs to stay home. We do have RT-PCRs for influenza as well, and it's commonly on respiratory panels. Respiratory PCR panels will test for 15 to 20 different respiratory pathogens all in one test. So influenza is always going to be one of those, and they will also be able to distinguish whether it's influenza A or influenza B. All of that data should go into national reporting, by the way. They need that data. So most hospitals will send that data up so it can be incorporated into the surveillance pictures. And then we can also do cell cultures. It used to be the gold standard where we would infect cell lines with influenza and look for cytopathological effects, CPE. We rarely do that anymore, but it is done occasionally and can be done. With all this testing, we don't need to go into this with a huge amount of detail, but your knowledge of the basic virology for these infections and when antibodies start appearing and are useful is always going to be important for when you want to order tests. If you want to order an antigen test, you just need to be aware of the fact that eventually those antigens get cleared after the immune system clears those antigens, so they become negative at that time. And you'll also get antibodies starting to appear, and then those titers over time will eventually diminish. Transitioning now, we're set up for Monday next week when we have lecture, but transitioning away from influenza into common cold viruses, you need to have a pretty good understanding of the differences between common cold and influenza. What's your understanding of like the major differences between influenza and common cold viruses? Okay. What else?
Who's gonna put you on your ass?
Flu will put you on your ass, yeah. Severe fatigue, yeah, exactly. In general, as a generalization, you get more systemic effects of influenza than you do with the more localized symptoms you see with common cold. Localized symptoms like sneezing, runny nose, maybe red eyes, things like that, stuffiness, sinusitis, those are more associated with common cold. Influenza fever, the body aches and malaise, that extreme fatigue where it just cracks you out, knocks you out. So it can be a little difficult to make an empiric diagnosis, but generally, those are the basis in which you can sort of get a sense of whether or not you have a common cold infection versus influenza. A couple of slides here just to help you get a clinical picture. We don't need to read through them, but put an FYI here about how a common cold would present, and the fact that you're still kind of semi-functional, you don't feel great, and you do have those localized symptoms, but you're probably not going to seek treatment. You're probably not going to get any specific antiviral meds for it. So maybe some over-the-counter drugs, analgesics, Advil, ibuprofen, things like that, cough suppressants, but nothing that would warrant you seeking like specific antiviral treatment. Contrast that with an influenza vignette. This, by the way, this is my sister-in-law. This is my brother, but put their names in there. Funsies. For this vignette, this is basically describing what would be a fully systemic influenza infection. So, exhaustion, too weak to get out of bed, you're staying in bed, high fever, 104 degrees Fahrenheit, calling your PA, asking for advice, coming into the clinic, getting a diagnostic test, usually a rapid test. It's positive for influenza A, which is typical with really severe influenza. And then if it's moderate to severe, then you could potentially get a prescription for Tamiflu, a specific anti-influenza drug, oseltamivir. Closing with that, something to just look at again. Note that neuraminidase inhibitors like Tamiflu or oseltamivir are sort of like the first line, front line drugs right now for treating moderate to severe influenza. Okay, that's probably good enough for today. Any questions? Okay, you guys are beat down today. I'm sorry with that. Good luck on your biochem exam, and I'll see you guys on Monday.